Well, you see, this is one of the developed labs picture image in front of you. You see how a lab is, developed, or you can say how a lab, is arranged. Basically, so conducting a good experiment means what? Why I have put this image here? Because to just tell you, the the research when you are doing now means what? Means if you are doing some work, all the successive stages of research equipment has to be present in one room. Okay, it doesn't mean that suppose I have done the autoclave here and shaking there and like a, PCR running somewhere else. No, a developed lab. I'm talking about since inception of this system. Okay, a developed lab means you must have all the devices and equipment at a one time at a one place where you conduct the research, and that is not possible in all the colleges. Okay, you see this image where everything is there. It's from very small thing, from start from incubator to the PCR, and everything is there. Well, again, you need some consumable thing which you use. Means these are the thing which we use in the lab one time, use and throw. We just use it and throw it, and that we cannot use it again. And these are basically small things, are plastic pipettes and disposal, this thing. Well, in case of weight lab, another big problem is that you need to maintain the data at every interval of your research. You just take the reading or calculate something and put it somewhere else and write it somewhere else. And unfortunately, if you miss this one, sometimes it happens. Suppose we have work on, like, we check. Suppose a very, you take an example of, like, microbiology. When you do some work in microbiology, suppose you start culture of microbiology in a petri dish. You can while culturing, you have used cup method infusion. Like, suppose you have kept some, like, you keep some antibiotic in the middle of, like, a petri dish, and you have checked minimum, yeah, zone of inhibition. You have to check zone of inhibition. Okay, how far the zone of inhibition is, inhibition is arise here. And by any mistake, suppose you have gone somewhere else, and somebody by doing some mistake, your petri dish another fell down, or suppose it's an it is a, get disturbed, or by any means it, like, you can say it vanish. So in that condition, you are a loser, because to culture any bacteria, to culture anything, you need a lot of time. And after spending a lot of time on a particular research, and when the time you need to take the reading or the result, at that time you are unable to get it, the reading or the result, because of someone's mishandling, or someone misbehavior, or someone's mis or malpracticing by you, and able to get the result. Even sometimes, as an we are human being, or you suppose you need, I know, okay, but by today morning I have to take this reading, and ultimately, I have seen that I'm get busy in some official work, and get busy in some official work, and suppose today onward there isn't a big leave in my college, and I'm gone to leave. Oh my God! When I come to my home, then I realize that, okay, I forget to take the reading, and a college is closed, and it will be closed for the 15 days and 30 days. So in that condition, the reading which has to be taken, or you can say, as it has to be or it's supposed to be taken by that time. You see, after 15 days, the rating will be different. So this is another big problem, to maintain the reading, to maintain the, like, a information proper way. Or if it's a difficult work, at the same time, when you're doing a work, On a wet lab, there is nothing to worry because everything is simultaneously stored somewhere where you want to keep it. That's it. Well, and again, in case of the wet lab, whatever the research you have, whatever the research you have conducted, whatever the research you have done, when your research is finished, when your research is ended, you have to expose, you have to dispose every, you have to dispose uh, every part of your research which need to be disposed after the research. You have to do it judicially. So these are the like in a suppose black bag, red bag, blue bag. These are the indicator in which bag you have to keep what kind of like a used research material. Suppose if it is a black bag, then it means that you have to keep your cytotoxic waste, expiry date medicines, radioactive substances waste. So these things you need to keep there. In other way, it's mean that what? It's mean that again a developed lab must have this type of uh, beans with a particular indicator where we keep our used research material accordingly. In a see just a red bag indicate that okay, we need to use the blood bag, we need to use the infectious IV set, infectious plastic tablets, rubber catheters, and all these things we need to be keeping red bag. In the same way, we need to keep something in a blue bag, like in a blue bag or blue beans, like in a glass bottle, the glass ampoules, and the injections while etc. So this is also a big problem. Problem is what? If you ignore to dispose your research uh, wastage in a proper way, it's been that somebody is going to be or somebody is liable to get infection from this by these all. Yeah, because you conduct, suppose if you conduct a research on like any rat and mouse or any animal, you have taken out the blood and you just recklessly you have throw the needle, you recklessly, carelessly you have throw the blood here and there, and we the human being we commit a mistake, or to err is human, and somebody use that needle by any means or any means, if that blood get inserted in body somebody else, 100% he will be liable to get the infection. Of you as a biological student, nosocomial, what is this nosocomial? When some sort of like an infection we get from the hospitals. So basically in the same way, in the big research lab, in a big research lab there are a lot of when you, I hope you have an experience still, I'm tell you, when you go to the big research lab like all CSR life labs, you see everywhere is a well developed like well protected door and shutters are there. Everywhere everyone is used a mask and proper scarf and proper gloves. you know they know because they know it's a very easy to get infection these in these lab on the hospital, because virus and bacteria are laying here and there. And as I'm telling you now, suppose it is our duty to dispose the part of the wastage part or the use part of the research which we have used it. But if you're not do it properly, means either you or somebody else are liable to get infection or is liable to get a problem. So this is also one of the responsibility which you need to perform as a researcher. Again here, when you're performing a dry lab research, nothing to worry. You don't need to do all this thing. You just sit in front of the computer and do your work.